In this video, we'll try and understand another important, or I would say a very important process of photosynthesis, that is chemiosmotic hypothesis or chemiosmosis. It is also said chemiosmotic theory. Now, this theory explains about the process of synthesis of ATP, which we have mentioned in cyclic and non-cyclic photophosphorylation. So both the schemes, that is whether it is cyclic or whether it is non-cyclic, it results into the production of ATP. So this theory help us understand that actually how the ATP is formed. Now, the process is known to take place in chloroplast and mitochondria. Both the places this process takes place. Now, when we are studying the process, let us try and understand the structure within the chloroplast. These are the two membranes, and these are thylakoid membranes, which have been labeled as outer thylakoid membrane and inner thylakoid membrane. So this is the inner thylakoid membrane and this is the outer thylakoid membrane. These two membranes are present and this is the structure of the thylakoid. Outside, what we see over here is the stroma, because the thylakoids or the grana lies within the stroma. So we have this structure. Now coming to the explanation part, let us try and understand there is a complex for splitting H2O, that is the molecule of water, or photolysis. There is a complex which is present on the inner side of the membrane of thylakoid. So there is a complex which is present for the splitting of water molecule within towards the inner side of the thylakoid membrane. Enzyme NADP reductase is present on the outer side, that is towards the stroma side or the outer thylakoid membrane. So these two structures are present, and remember, the complex for splitting water is towards the inner side, whereas the NADP reductase enzyme is present towards the stroma side on the outer membrane of thylakoid. The outer and the inner thylakoid membrane have internal space inside, which is known as the lumen. So this is the lumen over here. Now what we have studied, this we are talking of non-cyclic photophosphorylation. Now we have studied that the light energy is taken up by the photosystem 2, where the light energy is absorbed by the by the necessary pigments and are passed. This energy is passed on to the reaction center, which is chlorophyll A, and these are then passed to the primary electron acceptor, and through a series it passes, that is through the ETS or the electron transport chain. These electrons pass through the different pigments and different types of pigments and finally go to PS1, where again there is an electron acceptor. Now when this is passing through PS2, when the light energy falls on PS2, there is a primary electron acceptor, and then there are three other electron acceptor. Those are plastoquinone, then followed by the cytochrome, and also followed finally by the plastocyanin. So there are basically three members through which the electrons transport. This is the primary electron acceptor, and through the primary electron acceptor it first passes through the plastoquinone, cytochrome and plastocyanin. And again, from here, the electrons are passed to PS1, where again, and due to the excitation through the light energy and the electrons which are received over here, it again has a primary electron acceptor. There is another one for PS1. Now, we have also mentioned that during this transfer of electrons through the electron transport chain, during this phase which is marked over here, at this cytochrome level, there is a release of energy at cytochrome by these electrons. That is because the electrons are moving from high energy level to low energy level, they will radiate energy at the cytochrome. So that means when electron passes through cytochrome, cytochrome accepts a pair of proton from the stroma. So what is happening over here? As the electrons are passing from the acceptor, that is the plastoquinone, to cytochrome and plastocyanin, these are the part of the electron transport chain. Electron transport chain. So what is happening over here? At cytochrome, the electrons release energy, and this energy is utilized to pump the hydrogen, that is the proton, through the cytochrome into the lumen of the thylakoid membrane. These protons are situated or present in the stroma, and due to the release of energy as the electron pass from high energy level to low energy level, they will release energy, and this energy is utilized by the cytochrome to pump hydrogen, that is the proton, from the stroma to the inside of the lumen. So this is one source of energy, and here the cytochrome, although we say that cytochrome is an electron acceptor, it is also helping in the transporter, that is electron transporter, and it is actually helping in the proton acceptor. It passes the proton, allows the proton to enter from the stroma into the lumen. So therefore we have mentioned the cytochrome only over here in this particular diagram, and this blue is marking the movement of the electrons. Now, what is happening over here? Due to the continuous loss of electrons from this, there is splitting of water with the help of the complex which is present inside or the inner membrane of the thylakoid. So what is happening over here? The water molecules are splitted up to release 2 electrons. These 2 electrons will go to PS2. Then there will be a release of hydrogen molecule, that is the proton, and there will be oxygen. Now this oxygen molecule will actually is produced within the inside the lumen. So if you observe, the complex for splitting of water molecule is present towards the inner thylakoid membrane, and therefore the splitting of water takes place within the lumen or inside of the thylakoid membrane, and the electrons which are being produced over here are provided to the PS2 to continue with the process of photophosphorylation, because there is continuous movement of electrons from PS2 to the electron transport chain and finally to PS1. So this electron, these pair of electrons will replace the electron loss over there, and the hydrogen which is present over here and the oxygen. Now this oxygen actually diffuses out through the lumen to the stroma, that is towards the outside, and it is diffusing out through the membrane as a byproduct. The hydrogen is also accumulated within the, that is the protons are accumulated within the lumen. So as a result, due to the pumping of the electrons through the cytochromes, and also due to the splitting of the water molecule, there is a proton gradient developed. That means the lumen will now have continuously high concentration of proton inside as compared to the stroma, which is present on the outside. Two reasons very clearly: as the electrons pass from PS2 to the electron transport chain at the cytochrome, as the electrons are passing from high energy level or uphill to downhill, they will release energy at cytochrome, and this cytochrome as a result uses this energy to pump protons present in the stroma to the lumen of the thylakoid membrane. This is one source of proton. The second source is, since the PS2 system will continuously lose electrons, the complex for splitting the water molecule, which is present on the inner side of the thylakoid membrane, will help to split the water molecule, and this results into the formation of two, or that is a pair of electrons which replaces or which goes to PS2 and enters into the this electron transport chain. The hydrogen, which is the molecule of hydrogen, or the hydrogen which is being produced, is present within the lumen, and the oxygen which is produced due to the splitting is diffused out of the membrane as a byproduct. So remember, the oxygen is produced inside the lumen of the thylakoid membrane, and which later diffuses out to stroma. Now here, as a result of these two activities, there is high concentration of proton, or there is a development of proton gradient inside and outside. High concentration of hydrogen inside. Now, these protons will tend to come towards the towards the stroma. Now, these protons which are present because of the difference in concentration of protons, these protons will tend to come from high concentration to low concentration, and they can come through only certain channels of protein. So there are certain channels of protein through which only these hydrogen or these protons can come out to the stroma to actually balance the proton gradient. Now, there are specialized proteins, that is the F0 protein or F0 particle we call it. This F0 particle is an intrinsic protein. It is an intrinsic protein because it is embedded within the membrane. Now, this F0 is also known as integral protein because again it is present within the plasma membrane, F0 particle. It is having again another particle which is F1 particle. F1 particle are extrinsic proteins. Now, these extrinsic protein is actually ATP synthase, which is an enzyme, and it protrudes out from the plasma membrane of the thylakoid. Because it is present towards the outer side, it is known as the extrinsic protein. Now, the protons can come only through this channel. Now, what is happening over here actually, as protons move from high concentration to low concentration to these channels, they release energy. Now, as a result, if we mark over here, the proton will be entering over here with high energy, and it will release energy over here. This energy which is released by the proton, because what happens is the proton will bind to F0 and this will be passing through this and entering into F1. So here it helps, the F0 helps in facilitated diffusion. Why we are calling it as a facilitated diffusion? Because the protons are not simply passing through the membrane. Just as oxygen is passing through, the protons are not able to pass through the thylakoid membrane on their own. They require certain assistance, and that is facilitated by the F0 particle. Therefore, we call it as a facilitated diffusion. Also, you have to remember, because it is only the binding which does not require energy, so the diffusion which is taking place over here of proton is free of energy, or that means it does not require energy at all. At the same time, the hydrogen which is, or the proton which is passing through the F0, coming from high energy to low energy, it will release certain amount of energy, and this energy is utilized to bind adenosine diphosphate with an inorganic phosphate to form the ATP. Now, this ATP is therefore the result of the movement of the proton from the lumen to the stroma, and while coming, this releases energy, and this hydrogen that releases energy helps in combining ADP and inorganic phosphate to ATP, and the protons are then they come back to the stroma. So this results into the formation of ATP. Now, if we study over here again, try and understand the process. The other part, that is NADP, we'll deal it later. Try and understand the thylakoid membrane, the thylakoid which is present in the stroma or suspended in the stroma, has two membranes, outer thylakoid and inner thylakoid membrane. The inner thylakoid membrane has a complex for splitting of water, whereas the outer thylakoid membrane has NADP reductase, which is an enzyme. Now, what happens is, during the process, that is the photophosphorylation, over here we are talking of the thylakoid membrane, and also we will talk of the non-cyclic photophosphorylation. Over here, the PS2 system having the on receiving the light energy, the accessory pigments absorb the energy and pass it on to the reaction center, that is chlorophyll A, and from this the electrons are emitted, which are taken up by the electron acceptor, which we call as primary electron acceptors. Now, these primary electron acceptor then pass the electron to the series of, again, electron acceptors, the first being the plastoquinone, followed by cytochrome and the plastocyanin. And finally, these electrons are passed on to the PS1, where again the primary electron acceptor is there, and there is formation of NADPH from NADP. That we'll study later. Now, again concentrating over here this part, when the electrons are passing from plastoquinone to cytochrome to plastocyanin, at the cytochrome level, over here we have written in our non-cyclic photophosphorylation, that is ATP, ADP plus PI, giving ATP. This reaction we are trying to understand through the chemiosmotic hypothesis. So what is happening over here? When the electrons are passing from high energy level to low energy level, they will release energy at cytochrome. And this energy is used by the cytochrome to pump protons from present in the stroma to the lumen of the thylakoid membrane or the thylakoid. Now, within the lumen of the thylakoid, due to this pumping of a proton from stroma to the inner side, there is an increase in the concentration of protons within the lumen. Now, at the same time, due to the loss of electrons continuously from PS2 system, there is splitting of water molecules towards the inner side of the thylakoid membrane because a complex is present towards the inner side. So this complex will help in splitting of water molecules and it will result into the formation of two electrons, proton and the oxygen. The electrons are taken up by the PS2 system and they enter into the photophosphorylation. The protons which are being produced remain within the lumen. The oxygen which is produced as a byproduct moves from the lumen to the outside and that is towards the stroma. It is a byproduct. So remember that oxygen is produced within the lumen and the splitting of the water molecule also take place within the towards the inner side of the thylakoid membrane, that is towards the lumen. Now due to this reason there is an increase in the concentration of protons within the lumen as compared to the stroma, because from the stroma there is a continuous movement of the protons from outside to inside. Now these, because of the proton gradient, the protons will tend to move from inside of the lumen to the stroma, that is towards the outside. Now this can take place only through certain channels which are known as protein channels. Now these protein channels have two parts, that is F0 and F1. F0 particle being embedded within the thylakoid membrane, that is the outer membrane and the inner membrane, we call it as an intrinsic protein. Now this intrinsic protein actually binds up with the protons and when it binds, it releases energy because the protons over here are having high energy level. So they will release energy and pass on to the F1. Now F1 is an extrinsic protein because it is present, or extrinsic particle because it is present towards the stroma and outer side it comes out. It is not embedded, it is outer side of the thylakoid membrane towards the stroma, and it is also the ATP synthase. We are calling it as an ATP synthase, or it is an enzyme, because it uses this energy to facilitate or to help in combining the ADP with inorganic phosphate to form the ATP. And the hydrogen, that is the protons, are again produced or released into the stroma with the less energy level. Now over here you have to understand that when the, now coming to the next part, if we come to the next part, the electrons are continuously passing through that and they reach to PS1 system. When they reach to PS1 system, the electrons from the cytochromes to the plastoquinone, and from here they will enter the PS1 system. Now the PS1 system also has the primary electron acceptor, and at the same time we have also studied that this over here will form NADPH from NADP. Now, how this happens? The protons which are being released, that is coming from the lumen to the stroma, are taken up by the NADP. Remember, the NADP reductase enzyme is present on the outer thylakoid membrane, and the protons are also released, that is, they are also, the NADP reductase is also present towards the stroma side. The protons are always present in the stroma, which are able to combine with that NADP, and they get reduced to form NADPH. So here the chemoosmotic hypothesis help us to understand the formation of ATP, and this proton which is being released into the stroma due to this during the ATP synthesis is utilized for the reduction of NADP to NADPH, and these are important source for entering into the they are very much required for the dark reaction and are used to produce the or to further lead down to the process of photosynthesis. So we study that the chemoosmosis requires a proton pump which is being generated by the cytochrome and the splitting of the water, that is the proton gradient it creates, and also the energy and the enzyme which is ATPase, that is ATP synthase, which is required to create the ATP. Now, along with the NADPH which is being produced and also the ATP, the system of plant is ready to enter into the biosynthetic phase, that is the dark reaction.